the lecture on Chapter 9 and 10. Chapter 9 is relatively short and fairly easy, I think, compared to the last couple of chapters. They were fairly math heavy, and pH can be kind of difficult concept sometimes. So Chapter 9 is all about proteins, what proteins are, a little bit about their structure, and really builds on a lot of stuff that we already have as foundation knowledge, a lot of intermolecular forces we'll be looking at again. So this lecture really is a summary of Chapter 9, and then more detail about, and then really going through Chapter 10. Chapter 10 is all about enzymes. Enzymes are proteins, so they're proteins that act as catalysts. And they are very important in our bodies, particularly for metabolism, but really anything and everything in our body. There's thousands, probably millions of different, I don't know, maybe it's an exaggeration, chemical reactions that go on in your body that allow you to live, and they are all catalyzed by enzymes. So our introduction to enzymes will be in Chapter 10. Okay, so Chapter 9 is all about proteins. Proteins are important biomolecules. Remember, the biomolecules are proteins, carbohydrates, fats, and nucleic acids, which include DNA and RNA, which is Chapter 11, which we're not going to be doing this semester, but I encourage you to read it for fun because it's really interesting. But anyway, proteins are very, very important. They just like fats and carbohydrates, provide calories in our diet. So they do provide us with energy, and we will talk about that in more detail in Chapter 12 about how each of these different nutrients provides us with energy, or ATP. Proteins are important for playing roles in acid-base balance. We touched on this a little bit in Chapter 8 at the end. Because of they have an acid and a base. The amino group is a base. The carboxylic acid group is an acid, so they're like natural buffers. They are their own acid-base, conjugate acid-base system in a way. They make up components of the cell membrane. They make up transporters, things that let glucose in and out of the cell and other nutrients. They form antibodies, so they play an important role in the immune system because antibodies are very important in immunity, and they are composed of protein. And then finally, but not lastly, they form enzymes. And enzymes are biological catalysts. That would be the definition of an enzyme, is a molecule that speeds up a chemical reaction without being changed itself. And or I guess that's the definition of a catalyst, is something that speeds up a reaction but doesn't actually get changed in the reaction. And an enzyme is a biological catalyst. They are catalysts in biological systems, so in living things. So before we can really talk about enzymes, though, we have to understand a little bit more about proteins. So what are proteins? Proteins are made up of amino acids. So fats were made up of fatty acids and glycerol. Carbohydrates were made up of monosaccharides, usually glucose, connected in different, chains, different ways, with different linkages. Proteins are just connections of amino acids, long connections of amino acids. And just a reminder that we did talk about this a little bit in Chapter 8. This is what an amino acid looks like. There's a central carbon here, which is called the alpha carbon, and it has 4 groups attached to it, okay? It has the amine group, it has the carboxylic acid group, and as we learned at physiological pH, which is about 7.4, the amine group is protonated, so it has an extra hydrogen and it's got a positive charge, and the carboxylic acid group is deprotonated, so it is missing a hydrogen and it has a minus charge. So we call this a carboxylate instead of a carboxylic acid. When it's deprotonated, it's a carboxylate. So we've got the carboxylate group, the amine group, the hydrogen, and then the R group, which is known as the side chain. And that R group is different for each of the amino acids. So all the amino acids have the same amino group, carboxylate group, and hydrogen, and then they have a different R group, and that's what makes each amino acid unique. There are 20 different amino acids in our amino acid vocabulary, and you don't have to memorize them all. You would be given them their structures on a test. But you should understand the different categories of amino acids. So those 20 different amino acids can be grouped into the following 4 categories. The first one are the nonpolar neutral amino acids. So in green, highlighted in green, are the side chains of each of these amino acids. We've got tryptophan, phenylalanine, glycine, alanine, etc., etc. Notice that all of their side chains are mostly hydrocarbons, just carbon and hydrogen, carbon, hydrogen, carbon, hydrogen. Oh, there's a sulfur here, but that sulfur is not really very electronegative, so it's still going to be pretty nonpolar. The one exception, I guess, is tryptophan, which does have a nitrogen in the side chain. Nitrogen is a polar electron hog, one of the, what is it? Nitrogen, oxygen, fluorine, and chlorine are the 4 electron hogs. Okay, so normally having a nitrogen would make it more polar, but tryptophan happens to be in this nonpolar group because there's so many carbons and hydrogens otherwise. So these guys are all nonpolar because of their high amount of carbons and hydrogens in their side chains, and they're neutral, as in they have no charge. The side chains do not have a charge. The amino acids, the amine group and the acid group, technically still have a charge, but overall they cancel out. The amine has a plus charge, the carboxylate has a minus charge, so overall these amino acids still have 0 overall charge. They are neutral. I have a star here by proline. Proline is unique among the amino acids because it's actually, the whole thing is in a ring form. So for instance, phenylalanine here, the side chain has a benzene ring, okay, an aromatic ring structure, but the amino acid backbone itself is still sort of linear, whereas in proline, the amino acid actually, the side chain actually is bonded to that backbone. So where's the central carbon? Is right here. This carbon right here is the central carbon of the amino acid backbone with an amine group on one side and the carboxylate group on the other. So this is the side chain, but you can see the side chain loops back around and actually bonds back to that amino group. So it forms a ring. That actually affects the structure of proteins that have proline in their side chain. So it's unique. It gets a little star for being a ring structure. The next group of amino acids are the polar neutral amino acids. They all have polar side chains. Their side chains all have an electron hog, oxygen, nitrogen. Really, it's just oxygen or nitrogen in there somewhere. With the exception of cysteine down here, which has a sulfur group. This SH group is slightly polar. Sulfur is this sulfur carbon bond here is slightly polar. It's not a huge electron hog, but sulfur does hog more than the carbon does. Okay, but all these others you see, oxygen, 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 oxygen. Okay, these are all the polar neutral amino acids. And then you have the charged amino acids, which are the polar acidic amino acids and the polar basic amino acids. And they all have a charge. So the polar acidic amino acids have a carboxylic acid group in their side chain, which are usually deprotonated. So here's the carboxylic acid group in aspartic acid and glutamic acid. And in the polar basic groups, they have an amine in their side chain. So an NH or NH2 or NH3. Okay, so here's the amine in lysine, arginine, and histidine are the 3 polar basic amino acids. Okay. So those are the different groups of amino acids. When you form a protein, basically what you're doing is you're just making a chain of amino acids together. Chains of amino acids are proteins. And amino acids, when they're linked together, that bond is called a peptide bond. Just like when you had 2 monosaccharides linked together, We called that a glycosidic linkage. Okay, two amino acids together is a peptide bond. And just conventionally, when we write amino acids, when we're talking about the sequence of amino acids, conventionally you write them in this sort of format, N terminus to C terminus, okay? So it matters which way they're connected. So if you have alanine, valine, dot, 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 okay, that's different than having valine and alanine. They're not the same because they're different ends. So this one has the amino terminus on the left and the carboxy terminus on the right. You always write protein sequences N terminus to C terminus. So if you are, if alanine, I guess you could put it this way, if alanine's left hand is holding valine's right hand, it's different than if valine's left hand is holding alanine's right hand. It's slightly different. So you always want to write it with the N terminus on the left and the C terminus on the right. N terminus meaning the amino group, the nitrogen containing group, C terminus being the carboxy group. Okay, so let me just go back here and show you. This is the right order to write an amino acid. The N group is always on the left, the carboxylic acid group is always on the right. So when you put these chains of amino acids together, okay, so here's a string of amino acids, and this actually, I should point out, is drawn a little bit incorrectly because look here, they have the C terminus, the carboxylic acid group on the left, and the amino acid, or sorry, the amine group on the right here. So that's actually against convention. It should be flipped the other way. Anyway, protein structure takes place sort of in different levels. Okay, so when you take amino acids and you string them together in a long chain, that's really just the first level of the protein structure. We call that the primary structure of the protein, which is the linear sequence of amino acids. It looks kind of like beads on a string in this picture here. So when I say AAs, I'm talking about amino acids. So that's the primary structure, but then that long chain of amino acids actually does a series of steps of folding on top of itself. Okay, so the secondary structure of a protein can either take the shape of what's called a beta pleated sheet or an alpha helix. Essentially, this long chain, this like necklace, beaded necklace, can either wind up in like a helical fashion, like a phone cord, all coiled, or it can fold back and forth in a sheet-like fashion. So those are the two different types of secondary structure, alpha helix or a beta pleated sheet, okay? So then you take your big long chain of, you have this big long chain of amino acids, some of it's coiled in a cord, some of it's folded back and forth on each other, okay? And then you can take all those beta pleated sheets and alpha helices and fold them into some kind of three-dimensional knot. That's called the tertiary structure, the third level, the tertiary structure, also known as the three-dimensional structure of the protein, okay? Some proteins stop there. They fold up into their three-dimensional structure and they're done and they're ready to go and rock and roll. A lot of proteins are a little more complex, a little more complicated than that. They have multiple subunits, okay? So, for instance, hemoglobin is an example. And it's an example that's in your book. And it's probably this little molecule shown here. But it happens to have 4 subunits, okay? So basically, 4 of these things come together to form an even larger sort of complex. And then you have hemoglobin is complete. It's not complete without all of its parts, all of the subunits. It's kind of like, I don't know, building a machine, building a car or something like that. You have to form I don't know how to build a car. Building, whatever, I'm going to stick with car, okay? You have to have the wheels, you have to have the steering wheel, the seats, the doors, okay, all the parts have to come together to form the car. Each part is its own subunit, okay, but the car can't work without all of the parts being there. And same with a lot of proteins, a lot of enzymes. They have multiple parts. You have to build each part separately, and then you put it all together. So that putting it all together is the quaternary structure. Not all proteins have multiple subunits. Some are very simple, and it's just one piece, so it just has tertiary structure, the 3D structure. But if you have multiple parts, then when all those parts come, all those three-dimensional parts come together, those subunits combine, then you have the quaternary structure. Okay? The three-dimensional structure of a protein is critical for its function. All of the functions of a protein completely depend on its structure. So if a protein becomes unstructured or is structured incorrectly, it completely screws up the function of the protein. Okay? So there's a lot of different things that help to the protein to form this three-dimensional structure. So these three-dimensional structure determinants, as we call them, are basically attractive forces between the side chains. So you have all these amino acids in a row that all have side chains sticking off of them. And you can even see here in this alpha helix structure, these little red dotted lines here, these are hydrogen bonds being drawn to show you that that's how this chain actually, how that helical structure is held together. It's actually by these attractive forces between side chains. So side chains throughout this protein hold it in that three-dimensional structure. So the different attractive forces that are active in proteins are nonpolar or hydrophobic interactions, okay, also known as London forces, polar or hydrophilic interactions, so polar dipole-dipole attractions, hydrogen bonds, those would be your polar interactions. Salt bridges, which we'll talk about, are ionic interactions when you have two charged things being attracted to each other, and disulfide bonds. This is a type of covalent bond that can be formed between cysteine molecules that have this SH side chain in their side chain, and I'll talk about each of these on the next slide. So here we go. We have examples of each of them. It's a great figure from your book. So the first one we'll talk about are hydrophobic interactions, literally just London forces between nonpolar side chains. So here's two nonpolar side chains here. Both have just carbons and hydrogens, an aromatic ring. There happen to be identical side chains, but they could be two different nonpolar side chains. And they have that nonpolar, they're both nonpolar. And like dissolves like. Like is attracted to like, okay? So two nonpolar groups are going to be attracted to each other through London forces, which are pretty weak. So it's not going to be the strongest type of interaction or attraction, but it is an attraction nonetheless. They get close to each other. They are attracted through those London forces. The next one that we talked about was the polar attractions or hydrophilic, liking water, okay? So you have hydrogen bonds here. So there's two sort of polar types of interactions. So one is the hydrogen bond between amino acid side chains. So here's a side chain sticking off this part of the protein, another polar side chain sticking off this part of the protein. They both have OH groups. They both are able to hydrogen bond with each other. So you get a hydrogen bond here, which is fairly strong and helps to a fairly strong attraction that helps keep that protein sort of stable in that state. Another type of hydrogen bond that forms that helps to contribute to the structure of a protein is this one here. So I guess I should these polar side chains here bonding to water in the environment of the protein. So proteins are pretty much always in a water environment. Your body is 60-70% water. The earth is 70% water. Okay, water is everywhere. So usually proteins, whatever environment they're in, is going to be full of water. So water, they'll be floating around in water. And any side chains that are on the outside of the protein can engage in hydrogen bonding to water. Polar molecules like water. So they will want to orient themselves on the outside of the protein. Whereas hydrophobic or nonpolar side chains are hydrophobic. They don't mix well with water. Like dissolves like, remember the golden rule. So nonpolar side chains are going to want to orient themselves on the inside of a protein structure where they are away from water on the outside. Okay, so hydrophobic interactions always occur on the inside of a protein, and these hydrophilic interactions occur on the outer surface of the protein. They can also occur on the inner surface here, like between amino acids. Here's another example of a hydrogen bond occurring within the amino acid structure, holding the amino acid within the protein structure and helping to hold that protein in that three-dimensional structure. Okay, so those are some polar. We've got nonpolar interactions, polar interactions. We have salt bridges that can form between acidic and basic amino acids. So here's an acidic amino acid that has a carboxylic acid group in its side chain, a deprotonated carboxylate group, and a basic amino acid that has a protonated amine group in its side chain, NH3+. Okay, so that plus charge and that minus charge are electrostatically attracted to each other. Opposites attract, plus and positive and minus. So that actually forms a very strong bond, even stronger than a hydrogen bond because it's almost like an ionic bond. They don't quite form a bond, but there's a very strong attraction, like a magnetic attraction between the two. The strongest type of bond that holds proteins together and helps them maintain their three-dimensional shape are these bonds here, the disulfide bonds, okay? So this is two cysteines, a cysteine here and a cysteine here, and both of them have an SH group coming off their side chain. And when those SH groups get reduced, when they get oxidized is gaining oxygen or losing hydrogen. Okay, when they get oxidized, they lose their hydrogens, and they form this covalent bond between these two sulfurs. So there's actually a covalent bond there. These are not intermolecular attractions. This is an intramolecular attraction. We've actually formed a solid, somewhat permanent bond here. So disulfide bonds are very, very strong bonds. So if a protein has a lot of cysteines in it, it can form a lot of disulfide bonds, and that makes it very stable. Okay, so proteins, like I said, their function is dependent on their three-dimensional structure. So it's critical that proteins maintain their three-dimensional structure. But there's a lot of ways to destroy protein function without necessarily digesting the protein, without breaking the covalent bonds within a protein. You can break the intermolecular attractions because they're much weaker than those covalent bonds. And that process is called denaturation. So denaturation is just a word that means to unfold a protein, for a protein to lose its three-dimensional structure. So the key here is that peptide bonds do not get broken. So here's your amino acid chain, okay, that are all held together by peptide bonds in this long string of amino acids, okay? None of that chain doesn't get broken. The protein itself is still the same sequence of amino acids, but all of those different intermolecular attractions, those hydrogen bonds, the salt bridges, okay, you break all of those, the protein loses its shape, now it's denatured, it doesn't have the same function. Okay, so an example here is, so there's lots of different things that can denature proteins. Heat is one thing that can denature proteins. So in this example here, you see a raw egg versus fried eggs, and they look different. You know, here's the albumin, that sort of yellowy, clear part of the raw egg becomes white when you fry the egg. It's because the protein is actually, the albumin protein in that albumin is actually denaturing. It's changing its shape, changes its function, it changes its properties, so it turns white and hard. Acid Acids and bases can also denature proteins, can disrupt these intermolecular forces, these attractive forces. Even agitation, if you take this egg and you whip it really hard, it gets all foamy. That's how you make meringues or something. Again, it changes the protein structure. That fast mixing actually causes a disruption of these attractive forces. Okay, so those are the main things you really need to know about proteins. I want you to understand the different levels. I want you to understand what amino acid is and the different types of amino acids, different sort of classifications, polar, nonpolar, etc., based on their side chains. I'd want you to, if I gave you an amino acid, you don't have to memorize them, but if I gave you the structure of an amino acid, I'd expect you to be able to look at the R group and tell me which class it fits into, basic, acidic, polar, or nonpolar. And I want you to understand the different levels of protein structure, primary, secondary, tertiary, and quaternary. And I want you to understand this concept of how the three-dimensional structure of a protein determines its function and how denaturing a protein can change that function, and know what some of the different things are that cause denaturing of a protein. And then I also expect you to know and understand the different types of attractive forces that can hold a protein in its three-dimensional shape. Okay, the last part of Chapter 9, I think Chapter 9, Section 9.5, I don't touch on it all. Section 9.5 just goes through some example proteins. I think it talks about hemoglobin and keratin and collagen, or some of the above. Some different proteins in the body and how they can be denatured and mutated and whatnot. But I like to just skip that part actually and move on to enzymes and proteins function as enzymes, because the Chapter 9 talks a lot about them as structural proteins. So like keratin in your hair and nails is a structural protein, but some of the really key roles of proteins in life are as enzymes. So what are enzymes? Enzymes are, I'll read it even for you here, biologically active proteins that accelerate chemical reactions but are not consumed or changed in reactions. That is the definition of a catalyst. Enzymes are biological catalysts. They speed up chemical reactions. And because our bodies are made of water, okay, and so enzymes work in water environment, an aqueous environment, they like to fold up so that all of the polar amino acids are on the outside and the non-polar amino acids are on the inside. We just talked about that when we were talking about the hydrophobic and hydrophilic interactions. So hydrophobic interactions, afraid of water, take place on the inside. Hydrophilic interactions take place on the outside with water in the aqueous environment. So the environment that an enzyme is in actually helps it to fold into the shape that it's going to make. So a little bit of sort of terminology when it comes to enzymes. When an enzyme, whatever molecule that an enzyme works on, whatever molecule binds to the enzyme to have the reaction catalyzed, that molecule is called the substrate. Okay, so this chapter, I like how they do this. They pick a certain enzyme and they stick with it for the entire chapter and kind of use it as the sample enzyme for the entire chapter. And the enzyme they choose is hexokinase. Hexokinase is an enzyme that binds to glucose and also ATP, and then it speeds up this chemical reaction here where one of the phosphates from ATP is adenosine triphosphate, adenosine plus 3 phosphate groups, okay? It transfers one of the phosphate groups from the ATP onto the glucose. So here, your products of this reaction are glucose 6-phosphate, glucose, with a phosphate attached to the 6th carbon, and ADP, adenosine diphosphate, which has 2 phosphates, okay? And hexokinase here is that enzyme that catalyzes that reaction. Now we'll see this enzyme and this reaction again in Chapter 12 when we're looking at metabolism of glucose, okay? But for now we're just going to focus on hexokinase and how it functions in this reaction. So what happens is that three-dimensional structure we talked about, why it's so important as an enzyme, is because an enzyme has to fit its substrate. It has to, that substrate has to be able to bind to it, okay? So the site in the enzyme, which is usually like a pocket, is called the active site. That's where the substrate binds to, and that is where the catalysis actually occurs. So in hexokinase, here's a structural model of hexokinase, the three-dimensional structure, this green thing. And then you can see there's a little pocket here, which is the active site, and glucose is bound. It fits very snugly in this little pocket. The pocket is perfectly shaped to hold on to glucose. In fact, if you zoom in, you can see that all of the side chains, the amino acids that are in this active site, they all have polar side chains that can engage in hydrogen bonding with all of those oxygen groups and oxygen and hydrogens in the glucose molecule itself. Glucose is a polar molecule, so there's a polar active site that's set up perfectly to bind this polar molecule and sort of hold it there with these little hydrogen bonds. So hexokinase won't fit other molecules. Other molecules are either not polar enough or not the right size or shape. It's specifically designed to bind glucose and only D-glucose, because remember, enzymes are chiral specific. They only bind certain enantiomers. If this was L-glucose, it wouldn't fit properly. Oh, that's what I'm talking about on this slide right here, substrate specificity, okay? So for example, in this scenario here, let's say this is D-glucose. It fits perfectly into the active site, but L-glucose, the enantiomer, does not. It doesn't fit in the active site. So enzymes are very specific to their substrates. They're very picky. They only bind the one thing. They have one job and one job only, and they're very good at that specific job. So another thing this chapter talks about is cofactors and coenzymes, which I like to equate to enzyme sidekicks, okay? Like all the good superheroes have these sidekicks. Well, maybe not all, maybe just Batman. That's the example I chose here, okay? So Batman has Robin. And Batman could probably function without Robin, but, you know, Robin really helps him function. He's not terribly important. Batman's really like the main hero, but Robin, he probably maybe couldn't do it as well without Robin. So that's my lame analogy here. But anyway, enzymes have cofactors or coenzymes. They're pretty much the same thing. The difference being a cofactor is an inorganic substance, usually an ion, like a metal ion, like magnesium, iron, zinc, are all popular cofactors for enzymes. And coenzymes are small organic molecules. Most of them are derived from vitamins. So all those vitamins in your diet, vitamin B12, vitamin C, okay, a lot of those things, a lot of the B vitamins really, act as coenzymes. So what does this mean still? Coenzyme, cofactor, what's an enzyme sidekick? Oh, I don't have a good picture. Oh well, essentially let's well, I'll get to a better example later in the lecture, but essentially it's like an on or off switch. Like hexokinase has this little helper that binds to it and allows, in hexokinase's case, it happens to be a cofactor called magnesium ion. Okay, without that magnesium ion, hexokinase doesn't function properly. And cofactors and coenzymes actually help to regulate enzymes, turn them on or off if they're not around. The enzyme doesn't function, so it's one way to help regulate enzymes. But they also often help the enzyme bind their substrate, to fit their substrate. Okay? So when it comes to enzymes and their substrates, there's a couple of different theories about how they do that, how enzymes and substrates match. Okay, so there's the two theories, the two models are the lock and key model and the induced fit model. So the lock and key model is what it sounds like. You have a lock on your front door. There's one key that fits that lock that'll unlock it. If you try to put other keys in there, it doesn't fit. A lock and a key are made to fit each other. And you could have 100 locks and 100 keys, but only one lock is going to fit that, or one key is going to fit each lock. So in this model, the substrate fits perfectly into the enzyme. It's the only molecule that fits perfectly into the enzyme. That's the lock and key model. The induced fit model states that, like, when the proper substrate gets close to the enzyme actually changes its shape in order to fit the substrate, so that the fit is induced by the presence of the substrate, okay? And different enzymes follow different ones of these models, okay? So both of these models, I think, are thought to be correct, that some enzymes function using the lock and key model, some function using this induced fit model. Another thing to point out on this slide is some of the terminology here. When you have an enzyme and you have a substrate, they're separate, and then they sort of react to form what we call the enzyme substrate complex, also known as, abbreviated as ES, the enzyme substrate complex. That's when they're actually bound together. So here's another way to write that. Enzyme plus substrate, E plus S, becomes the enzyme substrate complex, the ES. Okay. Hexokinase, the molecule, the enzyme that we're focusing on in this chapter, binds the substrate glucose, and it happens to follow the induced fit model. So if you look here at this, all the green, the hexokinase here, when the enzyme is technically on its own versus when it's bound to the substrate, it sort of closes a little, kind of forms, kind of hugs the substrate a little. It gives the glucose a hug, so it does fold up a little bit. It is an induced fit. It's not a drastic change, but it does change its shape slightly when it binds to the substrate. So that's a little bit about enzymes and their substrates and enzyme a little terminology. So remember that glucose binds to the active site. This pocket here is the active site. And now we have an enzyme substrate complex. And we're going to talk more at a later point in the chapter about how the catalysis, the speeding up of chemical reaction, actually occurs in this active site. But first we're going to digress a little bit to understand how chemical reactions get sped up. First we have to understand how chemical reactions happen at all. Okay? And what we're going to talk about a little bit is something called thermodynamics. Excuse me. So when bonds are formed and broken, there's energy that's transferred in that. Okay, so bonds actually contain energy, and when you break them, you release that energy, and it takes energy to break chemical bonds. I said that wrong. It takes energy to break chemical bonds. When you form chemical bonds, you then capture energy in those bonds. So there's a lot of sort of energy involved. That's where the thermodynamics, thermo meaning heat. So chemical reactions require energy. When you do a chemical reaction, you are either breaking bonds or you're forming bonds, and either one, you're manipulating energy. So when chemical reactions occur, they occur usually, the sort of theory is that it occurs by having molecules basically crash into each other, and that energy that's caused by that crash fuels the reaction. So this slide is actually animated. So I'm going to go into slideshow view for a moment. Okay, so you have two molecules come in, they crash together, it causes the bond to break, and then you have these two, I have my thing? So you have these two molecules, they came in, they crashed together, and it broke up the yellow one and caused it to form a new bond with this little blue one. So now you have two different molecules on the other end. You have this one that has 3 balls and 1 ball versus the two that were two balls. Okay, that's my cartoon version of molecules, okay? But you see that reaction, they fly in, they hit, and then they form new bonds, okay? So molecules are flying around all the time. I mean, there's gas molecules in the air that are just whipping around, flying around your head, but chemical reactions aren't necessarily occurring. So why is that? Well, there's a lot of reasons why that is. One is that, you know, molecules can fly around and not crash into each other. Or they can fly around, they can crash into each other, but there's not enough energy in that crash to break bonds and then cause the formation of new bonds. Another thing is they need to usually collide in the correct orientation. So not only do they need to collide hard enough, but they need to collide in the right position. So the example I have here is this. If you have these two molecules, so this is two fluorine molecules together, so this would be difluoride, and this one would be nitrogen and two oxygens would be nitrogen dioxide, okay? If they crash in this orientation, this fluorine bond breaks and this fluorine-nitrogen bond forms, so what you end up with is a free fluorine ion and I don't know what you call this one actually because it's not a binary molecule, but so because they crashed in the correct orientation. In this one, this example down here, you have them crashing in the opposite orientation. So I guess this fluorine atom or molecule is crashing into the back side of the nitrogen dioxide, and the bond can't form that way. If you break this bond right here, this fluorine then would have to like jump around to the nitrogen on the other side, and that doesn't really work that way. So if the molecules crash in this orientation, no bond forms. If they crash in this orientation, then a new bond, one bond breaks and the other one does form. Okay, so orientation and speed are both important when it comes to chemical reactions taking place. Now, that sort of puts like a minimum on things when it comes to chemical reactions. There's a minimum amount of energy that's required, and there's a certain orientation that's required. So these different sort of requirements for a chemical reaction to take place are known as the activation energy. So I like to think of activation energy actually as incentive, the incentive for a reaction to take place. So what do I mean by that? I have this funny analogy that's funnier when I use it in class, but we'll try it this way just the same. So let's say we're in class and I say, Bob, Joe, I'd like you to stand up and trade shirts with each other. Okay, you're probably not going to do it because, I mean, why should you? I just told you to trade shirts. Why would you do that? That would make you very uncomfortable having to take your shirt off in front of the classroom. What if I said, I'll give you $5 to trade shirts in front of the class? You'd probably still say no. You'd say that's not enough incentive. If I said I'd give you $50 to do it, well then maybe you'd be like, okay, $50, I'd do it for $50. So $50, that's your activation energy. That's the incentive you need to do this reaction, which happens to be trading shirts, okay? And the reason that it took you some incentive to do that is because trading shirts would be somewhat embarrassing in front of a room of people. It would make you, that maybe not being in the other person's shirt, but that transition state between when you're wearing your shirt and when you're wearing the other person's shirt, that's kind of uncomfortable to be half undressed in front of a room of people. Well, the same thing happens with molecules undergoing chemical reactions. When this bond breaks between the two fluoride ions and a new bond forms with this nitrogen dioxide, there's a transition state. The molecules are somewhat half naked for a split second while that bond is breaking and the new bond is forming. And it's very uncomfortable for the molecule to be in that state. So in order for it to want to do that, you have to provide enough incentive. You have to provide a large enough activation energy. Okay? So that was going to be this slide here, sort of that analogy. So, and in a later slide, I'll show you actually chemically on the, if we could zoom in on the molecules themselves, what that transition state actually looks like as the molecule, some bonds are breaking and some bonds are forming. Okay, so when you have a reaction, like I said, this is all about thermodynamics, the transfer of energy or heat in a chemical reaction. There's always a change in the energy. When bonds are broken, that means a lot of energy is being used to break bonds. Bonds are formed. Energy is actually captured in those bonds. So depending on how much energy it takes to break bonds and the reactants versus how much energy is stored in the bonds and the products, reactions can either sort of result in a net gain of energy or a net loss of energy. So we have two different sort of classifications for chemical reactions. They can either be exothermic or they can be endothermic. An exothermic reaction, and these pictures here are what we call energy diagrams. So on the y-axis we have the amount of energy in the reaction, and on the x-axis we have the progress of the reaction. So we have reactants react to form products, okay? In an exothermic reaction, the energy of the reactants is higher than the energy of the products. So there's actually a net decrease in the amount of energy. Exo means like external, okay, so it gives off energy. There's a net loss of energy. Heat is released from the reaction. So actually, if you did an exothermic reaction in a beaker, you would actually feel it get hotter because it's releasing that heat. What is this bump here? What is this hill here? This hill is that activation energy, that incentive to get the reaction going, okay? Whether it takes $5 or $50 for you to want to change shirts, okay? That kind of a thing. So some reactions have very high activation energies. Some of them have lower activation energies. Excuse me. So this little hill here, all chemical reactions have an activation energy. They all have a little peak right away, whether they're exothermic or endothermic. So this endothermic reaction here has a big hill and then kind of drops off towards the product. Again, this is the activation energy. The activation energy definition, when you're doing an energy diagram, it's the difference between the energy of the reactants and the peak of this hill. This peak represents the energy of that transition state, that uncomfortable state. So the more uncomfortable the molecule is in that transitioning state, so I guess in my analogy, the more uncomfortable you are with your half-naked upper torso, the higher that activation, the higher this peak is going to be, okay? And the higher the activation energy will be. So again, exothermic reaction loses energy. The energy of the reactants are higher than the energy of the products. An endothermic reaction, endo means to take in, internal, okay? So endothermic reactions absorb energy. The reactants have a lower energy than the products. So heat is actually absorbed in this reaction. And again, the activation energy is the difference between the energy of the reactants and the peak of this activation energy hill, okay? So there's a lot of activation energy required for this endothermic reaction. However, so looking at these two these two energy diagrams, it looks like endothermic reactions require higher activation energy than exothermic reactions. But that's not always the case, so don't get that idea in your head. It's just drawn that way in this reaction, but there are exothermic reactions that can have a very high activation energy before it comes down. So it's not necessarily the case that endothermic reactions have higher activation energy, okay? So when we talk about endothermic reaction or exothermic reaction, that is referring to the heat of reaction. That's what we call it in thermodynamics. The heat of a reaction, is it exothermic? Does it lose energy? Or is it endothermic? It gains energy. So again, exothermic, the reactants are going to be higher than the products. Endothermic, the reactants are going to be lower than the products. So, sample problem. Which of the following reaction energy diagrams is A, an exothermic reaction, B, an endothermic reaction, or C, a faster reaction? Ooh, and I did not go over that yet. Okay, so first of all, which one is an exothermic reaction, A or B? The correct answer for an exothermic reaction, the reactants are higher in energy than the products. So B is an exothermic reaction. It releases energy. Endothermic reaction is A. It gains or absorbs energy. Okay. Which one do you think is a faster reaction? If you said B, you are correct. The amount of activation, so the activation energy, also helps determine the speed of the reaction, okay? So think of, it looks, just follow the way that the reaction diagram looks. If you were a person biking, okay, on a bicycle, up this hill, and then down this hill, if you were a person on a bike, and these were terrains, which one do you think would be easier to ride, A or B? Well, on this one, you have this little hill to go up, and then a big hill to go down, versus this one, you have a big hill to go up and a little hill to go down. So which one do you think you're going to bike faster? Probably this one with the little hill. The lower the activation energy, the faster the reaction goes. Okay, that's the rule. The lower the activation energy, the smaller the hill, and the faster the reaction goes. So which one is going to be a faster reaction? All right, this one has a big hill. This one has a little hill. Diagram B. This reaction here is going to be the faster the reaction because it has this short little activation energy, as opposed to this one which has a bigger activation energy. Okay? The lower the activation energy, the faster the reaction. You might even want to make a note of that. The lower the activation energy, the faster the reaction. So this brings us to how catalysts actually work. When you have a chemical reaction that's catalyzed, whether it be by some chemical catalyst or by a biological catalyst like an enzyme, the way that it speeds up the chemical reaction is essentially by making that activation energy smaller, making that hill smaller. So here's a sort of diagram of that. You have a reaction. Reactants are here and products are here. Is this an endothermic reaction or an exothermic reaction? Hopefully you said exothermic because the reactants are higher than the products. So it doesn't matter which one it is, but I was just trying to quiz you there. So normally this is the activation energy that's required for this reaction if you follow the purple line. If you add a catalyst, look what happens. The energy of the reactants and products are the same, but this activation energy, this hill, is drastically reduced. Okay? Enzymes reduce the amount of activation energy that's needed, so the reaction can occur much faster because it has a much smaller hill to climb. Okay? It provides an easier path for the little person on a bicycle. Okay? So how exactly does it do that? It's great to say that enzymes speed up chemical reactions because they make this little hill smaller. What does that mean that they're making this hill smaller? How do they do that? How do they change the activation energy? Well, they can do that a couple of different ways. And since we know what activation energy is caused by, it's caused by the activation energy is talking about how fast molecules need to collide. And it talks about how, and it incorporates the orientation they need to collide in, okay? So the more energy things need to have in order to meet that activation energy versus the orientation they collide in, enzymes can change those things. They can change those sort of minimum requirements. So these are the different things we'll talk about. Enzymes can speed up chemical reactions by altering the proximity, orientation, bond energy, and transition stabilization of substrates in a chemical reaction. Okay, so here's a good example. In the absence of an enzyme, you have these molecules, two molecules that you want to make them form a bond together. Okay, you want these these reactants, let's say A and B, to form this product here, AB. If these molecules are just flying around in space and they need to collide with each other at some speed in the proper orientation, what's the likelihood that that's going to happen? You know, what are the odds that they're going to find each other randomly in space? However, in the presence of an enzyme, when they're both attracted to this enzyme and fit nicely in this enzyme, it's a lot easier to get them together, get them close enough in order to orient themselves in the proper way to react, to form a reaction. So that's how enzymes speed up or sort of reduce the activation energy, because they increase or sort of optimize the proximity and orientation of two molecules so that that chemical reaction can actually form, increases the odds. The other thing that enzymes do is they can change what's called the bond energy. Okay, so here's a close-up view of hexokinase, that molecule, that enzyme we've been talking about the whole time. It takes glucose and ATP, and remember that it takes one of the phosphates off, or allows one of the phosphates to transfer from ATP to the glucose. So here's the products. Okay, what happens is when those molecules are in the active site, you have the cofactor, magnesium, which holds the ATP in place. And then you have a bunch of side chains that hydrogen bond with the glucose to hold it in place. And when you have those hydrogen bonds, think of them like little invisible tethers that are holding this glucose in place and kind of pulling on the bonds a little bit. Well, they actually are weakening the bonds. Same thing with this magnesium holding onto these phosphate groups. It's technically weakening this bond here between the two phosphates. So it allows it to break off and join onto the glucose molecule. Then what happens once the products are formed, the active site doesn't quite fit as well. They don't fit in the active site as well, because now they have a different structure. And remember, the active site is very, very specific. If the active site is not going to bind L-glucose versus D-glucose, it's not going to bind glucose 6-phosphate as well as it binds glucose. So once the products are formed, they don't fit as well in the active site and they're released and let go. So lastly, the last way that enzymes help to reduce the activation energy, and probably the most important way that they reduce the activation energy, is that they stabilize that transition state. So remember, in my analogy, where you're switching shirts with your classmate, okay, in front of the room, that transition state is that awkward middle point where you both have your shirts off and you haven't put the other shirt back on yet. You're sitting there half naked and everyone's staring at you and, you know, trying to find your 6-pack of abs or whatever, okay? So molecules go through the same thing. Here's an example from an earlier chapter. This is actually, if this looks familiar to you, it's from the reaction that occurs between like glucose molecules, monosaccharides, when they go from the straight chain to the cyclic hemiacetal form. Okay, so this is a sort of showing you in blue dotted lines the bonds that are formed in the product, and the red dotted lines show you the ones that are broken. Okay, so in this reaction, in order for these two molecules to combine to form this bond here, this is the transition state. Okay, so initially this bond forms and these two bonds break. Okay, so let's just draw that out. We have the new blue bond formed here, but this double bond has broken as well as that hydrogen has broken off. Okay, so in the transition state, we have this. Okay, these two molecules are very unstable. They're very unhappy like that. And they very quickly, this oxygen very quickly picks up this hydrogen and we form the product. But for a split second, a very tiny second, these two molecules are in existence. And in space, they would be very uncomfortable. But in that little active site of the enzyme, there's usually some side chains or something that can have some attractive forces that help to stabilize these two guys. Kind of like, if you want to go back to my analogy, it would be like if instead of saying you guys had to change shirts in front of the classroom, I would say you can go into a little tent, like a tent or something, like there's a space that nobody can watch you. So you have to change shirts in like a tent. That would be a little more comfortable. You wouldn't be worried about everyone sort of looking at you. And that's what the enzyme does. It provides sort of a private space for this transition state to occur. And that's really mainly actually how enzymes work to decrease the activation energy. They decrease the incentive because they make that a more comfortable transition state. Okay, so enzymes function to speed up chemical reactions by lowering the activation energy. And there's a couple different ways that they lower activation energy through helping molecules orient themselves, to helping them stabilize the transition state. Okay, and then I think this is the last part of the chapter. Factors that affect enzyme activity. So enzymes function best under certain conditions. And so measuring how fast an enzymatic reaction occurs is a measure of that enzyme's activity. And an enzyme's activity is optimal under different conditions. So some of the different things that can affect enzyme activity are substrate concentration, pH, temperature, and the presence of inhibitors. This picture here talks about an example that the book talks about. So if you have an apple, if you ever noticed this, if you have an apple, you cut it up into slices. I do this with my stepsons all the time. I can't slice an apple. If they're home, I can slice an apple and they'll eat it right then, fresh. They have no problem with apple slices. But if I slice an apple and I stick it in a Ziploc baggie and put it in their lunchbox, they won't touch it because by the time they get to lunch, the apple's all brown. The slices are all brown and they think it's gone bad and they don't like brown apples. Okay? The reason that the apple turns brown is because once you expose the inside of the apple to air, there's oxygen in air, and chemical reactions, oxidation reactions occur with enzymes, okay, that cause it to turn brown, okay? A little trick that you can do if you don't want your sliced apples to turn brown is you can actually treat them with acid. You change the pH and the enzymes don't work, so those oxidation reactions don't occur as quickly. So a popular thing to do to treat your apples with acid is to paint them, I guess, with some lemon juice. Lemon juice or lime juice will slow down the oxidation of apples. This is also a common thing for avocados, also brown very quickly when exposed to air. They oxidize very quickly, and that's why a lot of like guacamole recipes include lime juice, a squeeze of lime juice or lemon juice. It helps to slow the browning so it doesn't get brown. Okay, but we'll talk about why that is, what pH does. So the first thing on the list there was substrate concentration. So usually, if you have an enzyme, the more substrate you give it, the higher the activity is going to be. The more substrate you give it, the more product it's going to make. So if we look at this graph here, we're talking about the reaction rate, okay, how fast the reaction occurs, how much product is made, versus substrate concentration here, how much substrate is present, assuming you have a fixed amount of enzyme, okay? So you have a certain amount of enzyme. The more substrate you give it here, the more product you're making. But you see here at the top, you reach sort of a plateau. What is this plateau? We call this plateau the steady state of enzyme activity, okay? Basically what it means is that at some point you saturate the enzyme's ability. It's working as fast as it can, and it can't work any faster. So no matter how much more substrate you give it, it's not going to be able to work any faster. Sort of working at its maximum speed here. That is called the steady state. The book has this great analogy that I really like, okay? And it's, let's say you have these 3 workers, okay? They're your enzymes. They work at a bike shop. And their maximum output in an hour, let's say, is 25 bicycles. These 3 guys, given all their tools, okay? So you can think of the tools in the analogy are kind of like the cofactors or the coenzymes, okay? So the enzymes, the workers and their sidekicks here, can put out about 25 bicycles an hour. That's the rate that they can go at. So if you give them bike parts for 10 bikes, okay, they can only make 10 bikes. They only have bike parts for 10 bikes. They can make 10 bikes in an hour. If you give them enough bike parts for 25 bikes, well, they can make 25 bikes in an hour. But if you give them the bike parts for 50 bikes, they can still only make 25 bikes in an hour because they're still only 3 people with tools. They can only make so many bikes so fast no matter how many parts you give them. So that's what we're talking about with steady state and saturation. You have these enzymes. Initially, the more and more parts you give them, the more and more substrates you give them, the more products they can make. But once you reach a certain amount, their threshold, okay, they can't go any faster. So substrate concentration can increase the rate of the reaction, can increase the output to a point. So we're talking about factors that affect enzyme activity. Substrate concentration affects enzyme activity. More substrates means more activity, but to a point. There's sort of a threshold there. Another thing that affects enzyme activity is pH, the pH of enzymes' environment. Okay, so the thing about enzymes is they are most active at a certain optimum pH. So here's a list from your book of optimum pHs for selected enzymes. Pepsin is an enzyme in the stomach that helps break down proteins. It breaks peptide bonds. Okay, the pH in the stomach is about 2, so the optimum pH for pepsin is 2. If you take pepsin out of the stomach, or you put it in an environment where the pH is higher, for instance, as food moves through the small intestine, the pH gets higher and higher, and pepsin doesn't work as well. It works ideally at a pH of 2. Here's another example. Trypsin. Here's a good example. Trypsin here also is a enzyme that breaks down peptide bonds, but it's active in the small intestine because it likes a pH of 8. As the stomach acid moves through the small intestine, it becomes more and more basic because of bile and other secretions. So trypsin kind of has the same role of pepsin. Both of them break peptide bonds, okay? But they work at different pHs, and they work at different points along the digestive tract where there's different pHs. So the thing about pH is it's not like a trend. So with substrate concentration, as the substrate concentration goes up, the enzyme activity goes up. With pH, it's not like as the pH goes up or down, the activity goes up or down. With pH, it's like there's an optimum pH, and anything above that or below that, it's not going to function very well. So trypsin, if you put trypsin in an environment that's pH of 12, it's not going to work very well. If you put it in an environment with a pH of 4, it's not going to work very well. The reason is because when you change the pH, you can alter the tertiary structure, the 3-dimensional structure of an enzyme. You can cause it to become denatured because adjusting the pH can adjust the proteination of the different side chains of the amino acids. They can, based on the pH, they might pick up a proton or release a proton because that's what happens with acids and bases in different pH environments. So when it comes to pH and enzymes, the pH should be just right. If you change the pH from what the optimum is, it's not going to function as well. Okay? Same sort of thing when it comes to temperature. Enzymes are designed to function at a certain temperature, and any variation from that temperature, whether it be higher or lower, is going to affect the enzyme's activity, is going to sort of negatively affect it. So this sort of goes against common sense, I think, because a lot of times when we think of chemical reactions and things in molecules, when you increase the temperature, you increase the speed of molecules. So a lot of times we think increasing the temperature will increase the rate of a reaction. But that's not necessarily true if it's an enzyme, if there's an enzyme involved in that reaction. Because enzymes work, they function best at a certain temperature. So if you change it, whether you make the temperature too high or too low, you affect the function of that enzyme. So some good examples are here, a child with a fever here, okay? High fevers, one of the reasons they're actually so dangerous is because your body has all these proteins and enzymes, and if your temperature gets too high, it can actually denature proteins, denature enzymes, and they don't function properly. And you don't want enzymes that are required for vital functions in your body to become denatured. Fevers, however, are also adaptive, and the reason we get fevers when we're sick is because it's the body's attempt to actually denature the proteins of the invader, so the bacteria or virus. Your body, if your body temperature goes up, your immune system is hoping that you will denature some of those viral proteins, and then their enzymes won't work properly. So it can be helpful and adaptive, but it can also be dangerous if it gets too high. So again, most of your body's enzymes work optimally at 37 degrees Celsius, which is 98.6 degrees Fahrenheit, normal body temperature. Okay? So any variation from that, they work less optimally. The other example on the other end, when enzymes are too cold, okay, that's why we actually refrigerate a lot of foods that are prone to spoiling. What is spoiling of a food? Essentially, it's becoming contaminated with bacteria or yeast or something, some kind of mold. So if you keep it cold, it slows all of the growth of those organisms because it slows enzymatic functions in those organisms. So also even things like the apples going brown, okay, that's not from a bacteria, that's from an enzymatic reaction, an enzyme catalyzed reaction. You take an apple, you slice it, you put half of it on the countertop and half of it in the refrigerator, come back an hour later, the one on the countertop is going to be more brown than the one in the refrigerator because the cold helped slow down that enzymatic process. So that's why we refrigerate foods, to slow down enzymes and to slow down growth of organisms. The other thing that can affect an enzyme activity are inhibitors, okay? And there's different flavors of inhibitors. You can have reversible inhibitors and irreversible inhibitors. Okay, so inhibitors basically prevent the enzyme from forming an enzyme-substrate complex with the substrate. Reversible inhibition is when you can inhibit the enzyme and then that inhibitor can be removed and the enzyme can function again. So any reversible inhibitors usually bind to enzymes through some kind of attractive forces, like intermolecular forces, like hydrogen bonds or London forces, okay? Irreversible inhibition is when the inhibitor binds to the enzyme and then won't let go. It's like super glue versus magnets, okay? So an irreversible inhibitor usually becomes covalently bound to the enzyme, so it forms very strong bonds that can't be easily broken. So reversible inhibitors actually come in two different forms. We have competitive inhibitors and non-competitive inhibitors. So a competitive inhibitor is one that competes for the active site of the enzyme, okay? So you have your enzyme, and you have your substrate here, and you have your inhibitor here. So competitive inhibitors often they try to mimic the substrate. They look very much like the substrate. So here you have the substrate has this sort of like arrow with a square, and the inhibitor has like an arrow with a circle. Okay, so it looks very similar, and it fits very snugly into the enzyme. It tricks the enzyme into thinking that it's the substrate. Okay, so it'll bind, but it'll also unbind. So sometimes the inhibitor will be there. Sometimes the substrate can knock the inhibitor out of the enzyme. So the more substrate you have, when it comes to the competitive inhibitors, if you add more substrate, a lot of times having that extra substrate around can sort of bump out those inhibitors because it is reversible. They will bind, the inhibitor will bind, but it can also be bumped out by the natural substrate, which really binds it stronger. Okay, so an example here of competitive inhibition, and it's an application of competitive inhibitors in medicine, is this one, which is kind of interesting. So it's how methanol poisoning is treated in a hospital. So methanol is also known as wood alcohol. It is not for consumption, but sometimes people consume it. It has actually been used to spike. So if you buy ethanol for like research purposes for labs, back in the times of Prohibition actually, when alcohol was prohibited, a lot of alcohol, people would try to drink like laboratory grade alcohol. So what they did is they actually spiked laboratory grade alcohol, ethanol, with methanol to prevent people from drinking it because they essentially made it poisonous. Methanol, in your body, this is what happens to the ethanol and meth. So ethanol gets metabolized to acetaldehyde, which the liver can detoxify and rid the body of. But methanol gets metabolized into formaldehyde, which is very, very toxic to the body. It's essentially what you use to preserve like cadavers and stuff. So formaldehyde is very, very toxic and can kill you. So methanol is toxic. So anyway, people would drink ethanol that was contaminated with methanol, and they would get methanol poisoning. Or they were just really desperate for alcohol and just drank wood alcohol, methanol, not knowing that it was toxic. So if someone had methanol poisoning, one way to treat it was actually to give them ethanol, to give them pure ethanol, because both ethanol and methanol compete for the same enzyme called alcohol dehydrogenase. So if you drank methanol and alcohol dehydrogenase is converting it to formaldehyde, and this formaldehyde is poisoning you, one way to treat you, one way to prevent this reaction from happening is to outcompete the methanol by adding lots and lots of ethanol to overtake all this alcohol dehydrogenase and to hog the alcohol dehydrogenase, and then the methanol just gets excreted. Instead of getting metabolized, it just gets excreted. So that is sort of a real use of competitive inhibitors in medicine. The other type of reversible inhibitor is a non-competitive inhibitor. So a competitive inhibitor competes for the active site. A non-competitive inhibitor binds to a different site on the enzyme. So here's an enzyme, here's the active site where the substrate binds, and here's an inhibitor of this enzyme that binds to this sort of back door, all right? And when that happens, it changes the shape of the active site, so now the substrate can't bind, okay? So, but again, this is a reversible inhibitor, so this type of inhibitor can then unbind itself and make the active site available again for the substrate, okay? So we call this non-competitive because if you have this a bunch of this inhibitor, adding more substrate, you can't bump the inhibitor out by adding more substrate the way you could with a competitive inhibitor because they compete for different sites. So these non-competitive inhibitors are a little more, I guess, dangerous or detrimental to enzyme activity. Okay, so the last slide I have here is a sample problem. I like this problem because it's sort of real world. So lactase is an enzyme that catalyzes the hydrolysis of the disaccharide lactose into the two monosaccharides galactose and glucose. In the active site, it's active in the small intestine where the pH is around 6.8. And supplements of lactase can help people who are lactose intolerant digest lactose. Okay, so here's the reaction. Lactose, the disaccharide, gets broken down by lactase into galactose and glucose. The other facts we have in here is that this enzyme lactase is active optimally at a pH of 6.8 inside the small intestine at a temperature of what? 37 degrees Celsius body temperature, okay? So the first part of the question is, would the enzyme also be just as active in the stomach with a pH of about 1 to 3? What do you think? Hopefully you said no. No, it would not be active because the lactase enzyme is most optimally active at a pH of 6.8. pH of 1 to 3 is not a pH of 6.8. Doesn't matter if it's 1 to 3 or 11 to 13. If it's not 6.8, it's not going to be as active. So it kind of makes you think too about these pills, because you swallow them and they go into your stomach, right? And that would not be they're not going to be active there in the stomach. But once that enzyme moves into the small intestine, then it will be more active, or possibly the capsule degrades in the stomach and releases the enzyme in the small intestine. Part B. Would a lactase supplement act just as well on lactose if it's first stirred into cold milk prior to drinking? Do you think that this lactase enzyme would work as well in cold milk as it would in a body that's 37 degrees C? No, because the body temperature is when it's most active, which is 37 degrees Celsius. Cold milk is going to be much colder than 37 degrees Celsius, and the enzyme is not going to work as well. So the answer for both of these is no, because it's not the optimum pH or the optimal temperature. Okay, so this is the last slide I have. I did not work a lot of sample problems in this chapter, or in these two chapters, in 9 and 10. But maybe I'll record another video working some more sample problems to get you guys going with the homework. But let me know, as always, let me know by email if any of this material is troubling you, if you want me to work some of the problems, if you're having any problems on some of the homework problems, let me know, and contact your local tutors and get help with the material. This test that's going to be on chapters 9, 10, and 12 is going to be in class. It's not going to be a take-home open book test, okay? So you do need to make sure that you really are mastering this material and understand it and memorize it. Remember it, be able to remember it, okay? That's all. Thanks for watching. Bye now.